Hi, welcome to the Learn Adventures webinar series. Today we're going to be talking about how to access biodiversity data from the Global Biodiversity Information Facility, otherwise known as GBIF. In today's presentation, we are just going to start with a 20-minute, 25, maybe 30-minute presentation, and then we're going to leave the rest of this stream open for questions and answers. Now, GBIF is something that I use regularly. So in this presentation, what we're going to do is detail a general overview for what GBIF is. We're going to talk about how to actually access the biodiversity data, and then I'm going to give you a series of best practices for working with this data. So, hey, let's just begin. So first, what is GBIF? Well, GBIF is the Global Biodiversity Information Facility. It's an international network and data infrastructure funded by the world's governments aimed at providing anyone, anywhere, open access data about all types of life on Earth. So when we say all types of life on Earth and what information do they actually contain, we're talking about this type of data here. I realize I am now covering my own slides. So GBIF does have a wide variety of data. 3 of them are checklist data. So this is a lists of named organisms or taxa. This could be something like the birds of Guyana, or it could be the amphibians of Brazil, or the reptiles of Southeast Asia, right? So it's just a named list of saying these species are found here. Another type of data is called sampling event data. These are really detailed data sets where you can actually assess things like the community composition or the abundance of species in an area. This data is not the majority of the data on GBIF. There's only a few data sets that really have this data, but hey, if that's what you're looking for, GBIF might have it for you. Another big component of their data is the associated metadata. So this includes information such as taxonomy, the type of collection, so how was this data collected, the different locations, the media, issues, and flags. All of this associated metadata is primarily attached to the occurrence data. The occurrence data is what GBIF is most well known for. For example, this image right here shows the point occurrence data for red-eye tree frogs, right? This is really the location of an individual organism in time and space. Now, this is, of course, what we're going to be talking about. The other data sets, of course, are available. However, we are primarily going to be focusing on this occurrence data. So you may ask, where does this data actually come from? Where does all of this occurrence data originate from? Well, one of the biggest data sources is actually community science, like iNaturalist or eBird. The GBIF interface has already interfaced with iNaturalist such that research-grade observations are now available through GBIF, okay? So whenever someone messages me or asks about using iNaturalist data, I almost always point them to GBIF directly. This is partially because it is a data aggregator, so you will get that iNaturalist information as well as information from museums, as well as information from published studies or from organizations that have uploaded their data to the GBIF infrastructure. So again, this is a massive collection. I'm actually on the web page right now, there are about 2.6 billion, with a B, occurrence records on the GBIF dataset, right? So how can we actually use this data? Again, I'm gonna have to move myself out of the way. Probably what you'll see GBIF used quite a bit for is for species distribution models. So this is where species are found. This is probably which has the most documentation online. However, you can also use GBIF data for conservation assessments, for biogeographical studies, for gap analyses, such as where is there a dearth of research about a particular organism, invasive species management, as well as spatio-temporal trends. Of course, all of these intersect with one another and overlap in some way. And I've used GBIF data for my own master's research, which is going to take this GBIF data as well as information from the IUCN to inform and find regions of shared evolutionary history. I'm doing this particular with all of the reptiles and amphibians of Middle America. So that's, Mexico and Central America, parts of South America. And this is just showing that you can use this for a lot of different purposes, right? Okay, so here I have an itemized list, a step-by-step instructions for how to access this data. Now, of course, I can just tell you, hey, read this list, and, access the data. But instead, I am going to go to GBIF directly. Here we go. And we are going to do this process step by step. Also, I am working on a handout that covers all of these steps in detail so that if you miss a step, you don't have time to watch this video, you're in a crowded coffee shop and don't want to have audio on, you can access this data. So first thing you're going to have to do is go to GBIF, right? It's gbif.org, very simple URL, and you will need to make an account. This is really, really useful, and I promise this is going to be much easier than you would anticipate. So the first thing, once you have your account, is you just go up for here. I prefer to start by just going to Get Data. This is up in the top left. And then I'm going to choose that very, very first option, which is occurrences. If you are looking for specific data sets, there's an option here. But again, we are only focusing on the occurrences data set. So this is the main interface that you are interacting with while in GBIF. There are, again, 2.6 billion results, and we have a series of filters on the left-hand side. And then all of the occurrences are found here on the right-hand side. This first option is a table. You can see it as a table, or you can go to gallery, which if this has been uploaded from iNaturalist, for example, let's actually, hold on, let's go back to table. And we're going to do this with the red-backed salamander. So Plethodon cinereus. Forgetting how to spell it. Cinereus, is it with an A or a U? Boo, 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 boo. So again, I am streaming, so my internet might be a little bit slow while I'm doing this. Here we go. So if we type in the name of the species, we see we have around 350,000 results. From here, we can go to the gallery view, which, as I was mentioning, if this is from iNaturalist or a museum record and it has attached media, it will display here. If it doesn't have attached media, you get this little broken media link option. But if you were looking for just, red-backed salamanders, about 58,000 images are available. Another very useful view is the map view. So you can see that most of the records are from this area in the eastern United States. There are some records in other areas. We'll talk about that in a second, as well as a surprisingly high number of records in the western part of the United States. You can also look at the taxonomy. We won't go too in depth on these little metrics. But all this is to say that you can actually just see all of the data very easily on GBIF. But how do we actually get this information for our own research studies? The first thing we're going to need to do is use those filters. So we already have a filter enabled, right? We have a filter that is looking at one organism. What we are going to do is I'm going to talk about just a few of the most common data filters. There are a wide number of filters that you can utilize for your own data sets. This is just the simple view. The advanced view has even more different filters we can use. But in most cases, we are only going to need to work on the scientific name, which includes the taxonomy, the basis of record, and locations. These are really the main filters that are utilized. So let's say we want to go into scientific name, and you'll see that it's already breaking it down for us. So if I start typing in, let's type in Plethodon. I should have typed it into here because it's probably, see, I did spell it incorrectly. This is another huge use of GBIF because my spelling is awful. So let's add Plethodon cinereus into here. What you'll find is that it's adding the author tag. So this is the, Green 1818. That's the person who described the species originally. And we now have 143,000 results. So we had more earlier, probably because of my typo. If we go back to the map, we'll see a lot more records. So again, we don't see any more in the top left, which makes more sense given this species. But with the scientific name field, you can do the individual species, but you can also do higher order taxonomy. So if I wanted to just pull amphibians, I could type in class amphibia, and it will pull all of the amphibians. So use this scientific name. This is usually where you're going to start. You kind of already know what species you want. You can add multiple here if you wanted to, no problem. Another very, very, very common filter would be the basis of record. And here there are a few, here, I'm going to make this just a tiny bit bigger. This is about as large as I can get it. There are a few different bases. So we have human observations. This is primarily going to be things like iNaturalist, right? iNaturalist in this case, if you're working with bird data, maybe eBird, but these are human observations. Could also be from publications, right? However, there's also preserved specimens. So these would be ones that are in museums. Again, lots of data from museums are included within GBIF. I am so sorry. I meant to go over here and pull this over. Okay. So if you are looking at just human observations, if you want to see just the museum specimens, you can do that. Here, let's just choose human observations. Okay, perfect. Now, the last very common filter that we will talk about would be location. Now, location is an awesome field, and there's two ways of setting location. You can do administrative areas. We'll start there because it's a little bit simpler. Administrative areas are, you can think of them as political boundaries. These are geographic areas. These are essentially areas that have a name. So let's say we only wanted to see records that were in Canada, for example. So we're looking at the map view. We only want to see records that are in Canada. We can choose Canada, and it'll add that filter, and you see that now we only have the records in Canada. We don't have the records in the United States. We can also remove that filter if we want to, very, very easily. And then the other location, this is a really awesome tool. I love using this. This has been extraordinarily helpful for me. This is essentially where you can draw lines on the map and only get records from a certain area. So let's say I'm only interested in getting records from sort of this general area. I'll draw a very big polygon, and then you see this here on the map. We only get records inside of that polygon. So if you're working with a more amorphously defined area, this is really useful if you say a research station, which wouldn't be an administrative area, right? And you only want to get records within a single park or a single research station, a single area. This is the way you would do it. And once you've selected your filters, so here we go. We're looking at salamanders. We only want them in this area. The way to get the data, you may have already seen this. You just click this download button. Now, oh, here we go. We need to remove this from up here. So make sure that you are running it with just these fields. When you click download, I notice this is where a lot of people start getting confused because you have three different options. You have simple, you have the Darwin Core Archive, and you have a species list. For most studies, you will just need to use the simple data set. This includes the basic coordinates and some of the more common fields. Again, if you're just wanting to figure out where species are found, you probably just need the simple data set. The Darwin Core Archive is for more specific or more complicated questions. This will include that simple data, but it will include more fields. If there's any raw data that was uploaded about this data set, that will be included in there, as well as supplementary material and links to any metadata and media. So if you're working with iNaturalist data, for example, this Darwin Core Archive will have the links to all of the images so that you can very quickly go through and say, like, look at the images and do whatever you need to do with those images, right? I've seen this used for example, the cross-pollination surveys where they're trying to see what interactions are found through community science. You can say, find all the records of butterflies, click on each image, and then look at all of the images that have a flower, and then make your observation. So again, the Darwin Core Archive is usually for projects that need media or have just something more to it. And the last one is a species list. This is a very, very simple list of species. So in this case, our species list would just be this one taxa, right? Because we're just looking at one species. But if I had removed the scientific name, drew that area, the little location thing, and then said, hey, species list, it'll tell me, hey, here are all the species found in this area based on your filter. So this is really good. I see this used a lot for people who work for, say, a nature center or a particular region. This is how they can say, here's all the species that are found here, right? Super, super easy. Whenever you select whichever one you need, all you'll need to do is click on the little download button. GBIF does have this little display that will pop up every time, free of cost, not free of responsibility, essentially saying just make sure you are citing this data properly, which we'll talk about how to do in just a second. So once you click understood, it should be, here we go. There we go. Took just a second. So right now the data is under processing. It is preparing this data. I don't anticipate it taking too crazy long. But on this page, you will see quite a bit of features, quite a few different tools. First off is a DOI number. So if you're using this for a publication, this is what you'd cite. This is the DOI number. It even gives you the citation right here. This ensures that anyone can find your DOI number and look at the exact filters you used to download this data. Because here we have the filter that was applied. This is the actual filter that I've used. And what is awesome is you can rerun this query. So say you have a query that you set up. It's a year later. You want to get the most up-to-date data. You just got to hit rerun query, and then you get it. So these downloads do take a while. Of course, everyone is working with it. The best part is once you have your account, GBIF will just email you saying your data is ready. And if we go to my account, we can actually go to my downloads section, and you can have a full history of all of your downloads that you would ever need. So this is the one, again, that we are downloading. This is the most recent one, but I have many different data sets. Some of these are just for fun. This is me looking at all of the amphibians and reptiles of Belize, but I also have one for my master's data. This is just the amphibian data set, so about 900,000 records. I believe my combined data set was around 2 million with raw records. But that, in essence, is how you actually just download the data. So what I'm going to do is specify in these next few slides, which would be here, a quick guide. So these are the quick guide and the data filters. We covered this we just covered this, so we don't need to go through these again. The basis of record. Coordinate uncertainty is something I wanted to talk about, but I realized it's better at a different section. And again, here are the geographic constraints. So you may have noticed that there were many issues and flags. So with any data, you're going to run into data issues. This is something I need you to understand about GBIF. GBIF collects and distributes records. They do not clean and remove most records. They will remove records on occasion, but it is very rare they do so. This is because GBIF prefers to highlight potential issues, leaving it up to you to decide how to handle them. This is a very, very logical way of handling the data. Again, they have 2.6 billion records. Manually validating all of those would be awful. And the problem is that every project has different needs. One person's junk data is another person's perfect data. You might find records that are missing, that have terrible accuracy problems, right? And you may say, this is worthless. I can't work with data that has, 2 or 3 kilometers of accuracy. For someone else, that may be perfectly fine. And so instead of GBIF deciding that this doesn't meet the thresholds, they're going to let you decide it. Ultimately, it is your responsibility to clean the data. And there are a wide variety of issues with records. I'm going to talk about 3 common ones, but again, there are many. Luckily, GBIF does flag all of the issues. In fact, if we go back to where we were on GBIF, here, let's change this view really quickly. Just for this data set, we can see right here, issues and flags. We can see all of the different possible issues here. Okay, some of these are not really issues. So the continent is derived from coordinates, right? That's just saying that, hey, we didn't know the author, whoever published this data, did not put the countries. We figured it out from the coordinates. Okay, we just assumed. But other ones, maybe that the coordinates are rounded. Okay, and we'll talk about a few of these issues very, very quickly. First, we'll just talk about geospatial issues, right? So coordinate invalid means that the coordinate falls out of the natural range, right? The latitudes and longitudes are either, well, they're either missing or they're outside of the 90, -90, 180, -180 range. A lot of the times this is just from a simple typo. Someone put 1.8 instead of, or well, I guess that someone put 1800 instead of 180, right? Or someone meant to put, -2 and then they put something that doesn't make sense, or they have flipped the coordinates around. They've put latitude where longitude needs to be and longitude where latitude needs to be. So that's all this is saying. And the best part is, because they just flagged it, you may be able to look at it and say, oh, yeah, they just forgot to add the negative sign. And so that's why it's in a completely different area. So keep that in mind. Country mismatch is because the data was uploaded and it said, in this case, that the record was found in Mexico. But in reality, the coordinates are saying that it is not in Mexico, it's in the United States, maybe in San Diego. This could be an accuracy issue. This could just be a coding issue, like they inputted the wrong country. But just keep that in mind, that the interpreted country and the country code are not equal. This could be a problem. This could not be a problem. Another one is accuracy. Accuracy is a very tricky one. So this includes things like coordinates are rounded. The accuracy is not a feasible number. You will occasionally see accuracy values of 0, which is not feasible, because there's always a little bit of an accuracy with GPS coordinates. But this could also be that the accuracy is rounded. We'll talk about that in just a second. The next one I want to talk about are taxonomy issues. One of them could be that the taxon doesn't match the higher rank. This essentially means that we have a species, we say it's this species, but it doesn't actually match with the family level designation or the genera level designation. This is just saying that there could be new taxonomy. This may have not been updated yet, or it could be that this is the wrong species and the other information is correct. So just, look at these issues and figure out step by step what might be the issue. Another one is the taxon ID issues. GBIF has one of the most robust taxonomy backbones. I think really anywhere for taxonomy, GBIF is one of my favorite places to go to. And so if there's a taxon ID that is the unique identifier for every single taxonomy, if it doesn't line up with the GBIF backbone, that's what they're saying. That's saying we don't know where this record is supposed to belong to. Again, could just be a minor error, something that you may not need to worry about. So date issues is another very common issue. This could be improper format, missing parts, not following standards. If you've worked with date data in any capacity, you know how frustrating it can be just from people using year, month, day instead of year, day, month, or vice versa. It could be someone putting it in a different format. It could be someone having a typo, right? The month is 16, but there aren't 16 months. So this is just saying that the date itself is broken. So just keep that in mind. Now, we're going to talk about a few best practices, okay? And as much as I wish that I could really do a deep dive into this data in this webinar, we just simply can't. So first best practice, clean your data thoroughly. Like we said, you need to clean your data. GBIF is not going to clean it for you. Unfortunately, there have been several published studies that did not mention in the slightest how they clean their data, and you just kind of hope they cleaned their data. So this means removing outlier points. You can very easily just plot these on a map. Using that location tool that we used, you can constrain the points to a known geographic area. If we're looking at those plethodons, those little salamanders from the eastern US, it doesn't make sense if there's records in Africa, right? Those could be invasives. Those could be improperly coded GPS points. They could be any number of things, but make sure you're removing them. If you're working with a lot of occurrences or a lot of species, I really like using kernel density estimates. They've helped me tremendously. Again, a little bit hard to explain here, but just make sure you're removing outliers and double check that taxonomy. Taxonomy updates all the time. You need to make sure you're checking it, even if it's just a manual look through. Unfortunately, it kind of does suck to check taxonomy. Use a mix of cleaning before and after download. This means that you're using those data filters to condense your data set down into something more manageable. And then within R, within Excel, within Python, however you want to clean your data, you are cleaning that data. Now, of course, like I mentioned, how to clean occurrence data requires a much deeper dive. There's a lot of nuances within it. I really recommend using the Coordinate Cleaner R package. That's with two capital C's. It is awesome. It'll do the very quick data cleaning. It's usually the first step I run it through, and there's a wide variety of issues. I highly recommend it if you're just wanting a quick cleaning of your data, and you can wash your hands and say, I don't need to look at this anymore. Coordinate Cleaner R package, awesome, awesome, awesome. We're going to move on to the best practices, and this is where I wanted to talk about accuracy. Accuracy is a very, very tricky metric to properly work with. Because the first question you need to ask yourself is that accuracy intentional? GBIF actually has a lengthy guide of best practices for how to round or hide your GPS points due to the taxa being threatened. They actually recommend if the taxa is at a very, very high risk of poaching, for example, to not even publish the data. But they have a sliding scale of other ways you can help obfuscate these records. One of them is just rounding the data. So you'll see that one of the biggest issues with that salamander data was that the coordinate was rounded. This may be because it's on a protected area, or it could be, not necessarily in the case with these salamanders, but say with elephants, right? They may have rounded the data to the nearest degree. So the accuracy could be saying that it's super, super high, right? But that might be intentional, okay? But then also ask yourself, what accuracy threshold is needed for your project? We kind of touched upon this. If you need very fine-scaled accuracy, then that's your accuracy threshold. But if you're just looking at if you're just looking at the images, right, and the exact location isn't necessarily relevant, you may have a much higher accuracy threshold that you can utilize. There is no standard, there is no best accuracy that you should use. It really depends on your study. Oftentimes, I just look at a map and I say, this point, if it was an accuracy of 100 meters, would that really mess up my data, or is that fine? So again, this is where the knowledge of your system and your study species comes into play. And the last best practice I really want to touch on is biases. We really need to understand biases in our data. One is just an observer sampling bias, right? It is much easier in the United States, is where we'll use this example. It's much easier to find a squirrel than it is to find a salamander, right? So naturally, there's going to be more observations, particularly from records like iNaturalist. There's also something, I'm just going to call it the country with attention bias. And I think that's really best exemplified by this image here. This is looking at Hylidae. So that's just like your tree frogs, right? And you'll see a huge number of records. And then around Nicaragua, not much. This is a very common issue if the countries or really just regions with attention, if they are not conducive to performing research, if they have not had the same amount of attention. For example, Costa Rica may not necessarily have more tree frogs or more species of tree frogs. They just have more attention. So that's why there's more points and larger points in Costa Rica. They do get a lot of research attention, right? So just keep this in mind that we're not working with abundances. We're working with presence data. That's sort of the third point. This is not presence-absence data, and that is a very tricky nuance to really wrap your head around. We are only recording with this data where a species was seen. We are not recording where it is not seen. Okay? So think about it. If you're going out into the woods and you go out to the same forest every single day, you sample every single organism, there's still probably some species you have not seen yet. And that is the same concept with this. Just because it wasn't noted there doesn't mean it isn't there. That is a nuance that is particularly for very specific methodologies. And there are some data sets. There are some data sets in GBIF that do include absence. That would be ones that need that Darwin Core Archive, the ones that have the published studies. But for the vast majority of records, it is presence data. And so with that, I want to say thank you. I want to open this up for questions. We were right on time. There will be another webinar next week, same day of the week, Tuesday. I'm going to push it forward by 2 hours, just to better account for everyone. But I really do want to just field any questions. We can go deeper into GBIF. I am working on a handout, actually several handouts. Let me show you those really, really quickly. Here we go. So I am working on several handouts that are including things like accessing GBIF data, this is very rough, quick guides on the filters and how to handle common issues, as well as some best practices. So whenever this webinar recap is over, I will be posting those. You will get an email with a link to this webinar as well as these handouts. So trying to make this as conducive for you, as well as the slides that I've produced. They'll all be in one big zip file package. But yeah, that was our wonderful first webinar. So if you have any questions whatsoever, if you just want to, and they can be about anything. We have the time. I'm giving you the full hour. I can now finally look at the chat. Ooh, Kazakhstan. That's a cool one. I was actually supposed to go there. And then the trip fell through. And yeah, that we were supposed to do some bat and herp stuff. That was going to be a fun one. And yeah, no, I love doing this. GBIF is a very big passion of mine. I use it a lot. Is there a method to request more precise to obscured species? Sorry for the INET lingo. Yeah, so that's one that oftentimes it is not very clear in the documentation if GBIF gets those obscured records. Unfortunately, in most cases, you have to email the observers directly or you have to send in a special request. So that's in the case of working with very specific data sets. You may have to send in a specific request, but I'm not sure. I do need to double check how they handle the obscured records, because it may just be the accuracy issue, right? They just increase the accuracy threshold. But I know with like iNaturalist records specifically, I've had people just reach out to me, DM me directly to get those records. On iNaturalist, you can also request to become they have a terminology. I think it's like a trusted data user or something, and then you can access those obscured records with higher accuracy. Yeah, I know it's so tricky working with threatened or hard to find species. Mainly just getting the data, it seems like. And I realize I didn't properly mention it, but next week's webinar will be visualizing your data with ggplot2. If you have any other types of stuff that you want. Curious to know which organization manages the GBIF database and how they pull data from the various sources. So the organization itself is GBIF. So that is their whole organization. Here we go. Let me change this view. Yeah, so it is it's many organizations, I guess is the more technical. So GBIF is the larger umbrella organization. They are based in Copenhagen, but they have many, many different people working on this. There's a governing board. There's lots of institutional help. GBIF itself does have fairly good resources. But you can see that they work on informatics, data products, participation. How they pull that data, a lot of it is reported to GBIF directly. So there are standardized data reporting features. Some journals, if you are submitting occurrence records, will automatically upload them to GBIF. I had that for one of my papers in Checklist, where they asked me to get it properly in format for upload to GBIF. For things like iNaturalist or eBird, those are just piped in, so I believe it's an automatic process. So I've seen GBIF records from iNaturalist come up very, very quickly. Can you suggest some small-scale project ideas to start exploring GBIF? Oh yeah, absolutely. So when I was learning how to do spatial analyses and how to do my own data, I just basically screwed around in GBIF. So I will say it depends on what you are looking to do. The project I did to figure out just how to work with these data points a little bit better was an analysis for how amphibian species ranges overlap with protected areas, right? So I just downloaded GBIF data, downloaded a series of protected areas, and just looked at how they went together. And actually, in some past webinars, I used GBIF data actually, for I think it was an Intro to R webinar. We used GBIF data and we combined it with a data set for AmphBio. So that's just saying like how big organisms are, how much do they weigh, stuff like that. And with that, so we have, all the amphibians of North America, we have all of their sizes, their average sizes, right? And we could see maybe an example of a Bergman's rule, right? Are species bigger at higher latitudes, right? So you can do lots of different projects like that. If I have to give some other ones, species distribution modeling is very, very, very useful to learn. Maxent is probably the first one you'd want to learn. That's a little methodology. So yeah, you can do just overlaps. You can play around with it. You can attach it to other data. That's one of my favorite things. And in fact, if I am allowed to plug myself, the webinar will be posted on the library that I have up on my website, learnadv.com. And I will say that the library got really weird in the last 2 days, and I'm not really sure what happened to it, but I have an open access database of open access data. So GBIF is on here, actually, but you can pair it with other data sources once this thing loads. Here we go. So for example, if you want to add open traits to GBIF, I can zoom in just a little bit more. You can compare it to climatic variables like WorldClim. You can add, here's that protected areas database I mentioned, as well as a few other areas. So if you want to add stuff together, you absolutely can. And do a similar session for how to use eBird data for research. Yeah, I want to use one specifically. eBird's a bit of a trickier one that I'll need to do a little bit more research on. I primarily work with occurrence records, so GPS, it was found here and there. But I know eBird has some other data sources that you can use. But no, we can absolutely do that. Let me add that to my list specifically because I know a lot of people want that. So eBird data. Okay, perfect. So yeah, I've added that to my webinar list. Let's see here. Let's